Originally, scientists thought that proteins were the genetic information that was passed on through generations. It's pretty understandable because they didn't even know that much about DNA back then. Today, even first graders know what DNA is. Today we're going to walk through the history of how DNA was discovered as the genetic material of organisms. In 1927, a scientist named Frederick Griffith performed this experiment on bacteria. He noticed that a substance from the harmful bacteria transformed the harmless bacteria into ones that could be unsafe. He called this bacterial transformation, showing that there's a heritable material that organisms have that can be passed on from one to another. I actually have a video on the Bacterial Transformation biology lab. But anyways, in 1952, Hershey and Chase came up with evidence to prove that DNA is, in fact, the genetic material, not proteins. They labeled some bacteriophages with radioactive sulfur and others with radioactive phosphorus. Keep in mind that proteins have sulfur and not phosphorus, and DNA has phosphorus and not sulfur. So they allowed the bacteriophages to infect bacterial cells by letting them insert their genetic material into the cells. After that was done, they separated the cell parts by agitation with a blender and a centrifuge, which is a tool that can separate mixtures. The result looks like this. All of the bacterial cell parts form a pellet at the bottom of the tube, and the liquid bacteriophages are up on top. In the group with radioactive sulfur, radioactivity was detected in the liquid. In the group with radioactive phosphorus, radioactivity was detected in the bacterial cells themselves. And since the bacteriophages had inserted their genetic material into the cells to infect them, Hershey and Chase knew that DNA had to be the genetic material since it has phosphorus, and radioactive phosphorus was detected in the cells. A little bit after the Hershey and Chase experiment, a woman named Rosalind Franklin carried out an X-ray crystallography analysis of DNA, showing that it had the shape of a double helix. That's why I always draw DNA like a twisted ladder. Here's the actual picture of what Rosalind Franklin saw. Also, X-ray crystallography is when an X-ray is aimed at the crystal of inspection, and it creates a diffraction pattern. The pattern that results is what's in the picture here. What's really sad is that two other scientists, Watson and Crick, proposed the double helix structure of DNA based off of their observations and won a Nobel Prize for it. Rosalind Franklin had already died when the prize was given, so she did not receive it, although her work was critical for the discovery. The reason why the structure of DNA is so important is because it has a central role for how it replicates itself. I'll be doing a DNA replication video soon, so keep an eye out for that. Finally, Meselson and Stahl carried out an experiment that showed the semi-conservative way DNA replicates. This means that one strand of the old DNA is always seen in the new DNA. Check out how this strand replicates. First it's separated, and then new nucleotides are added to match up with the old nucleotides on the old strands. What Meselson and Stahl did was grow bacteria in a heavy nitrogen isotope first, then transfer them to a lighter nitrogen isotope. After generations of letting the DNA replicate, they observed that the DNA looked like this. One strand was from the heavy isotope and the other strand was from the light one. This could have only happened if each DNA molecule had one strand that was from the parent molecule. Therefore, Meselson and Stahl concluded that DNA replication is semi-conservative. And that's where the history ends. Thanks for watching, and be sure to check out my other videos.